Good morning, everybody. I'm Audrey Tremoulet from UC San Diego, and I have the pleasure of sharing the podium this morning with three of my colleagues from UCSD. So I'm going to give you just a couple of slides to introduce our team, as well as one of the support pool projects, and then I'll hand it off. So just a couple of disclosures that are listed here, just to be transparent, although these are not really associated with the projects that we're doing generally. And this is the esteemed team of colleagues that I have the pleasure of working with in MPRINT at UCSD. Today on the podium, we're going to be joined by Dr. Christina Chambers, who is a PK, she'll be discussing PK of antibiotics in breast milk and milk composition and antibiotic-treated mothers. That will then we'll have George Liu come up, who is our division chief of Peds ID, who's going to be discussing effects of antibiotic exposure in breast milk in a mouse model. So we have the basic science component. And then Shirley Tsunoda, who oversees the data science project with Peter Dorstein, is professor of pharmacy, and she's going to be studying the impact of antibiotics on infant metabolome. So I want to just give a moment of podium time to one of our support pools, which is led by Dr. Pia Panaraj, who's a pediatric infectious disease physician who was with us originally as a resident in pediatrics, went on to do her Peds ID fellowship, and we were lucky enough to be able to recruit her back to be with us. And this is currently being supported by one of our support pool projects. And she's working on the impact of maternal milk IgA binding to fecal bacteria on oral rotavirus vaccine in response in infants. And the really lovely thing about her project is that it starts to move into the world of microbiome, which we have a big emphasis in at UCSD, and we hope to be able to have even more overlaps there in MPRINT eventually. The hypothesis here was that the non-rotavirus specific IgA in human milk is associated with the infant gut microbiome composition through IgA binding to fecal bacteria. And she's also looking at the immunological responses and frequency of Tregs, as well as the oral vaccine response. And her aims were to determine the proportion of IgA bound and unbound bacteria transferred from human milk to infant stool, as well as to determine the relationship between differential IgA binding and the rotavirus vaccine serologic and T-cell responses. So currently, this is just at the beginning stages, and I'm hoping that she can join us next year and kind of giving some updates. But so far, she's collected mother's milk and infant stool from 60 rotavirus vaccine responders and non-responders. She's also set up the spectral flow cytometry panel for deep functional profiling of the T-cell responses, and she's working on early analysis. And she's currently optimizing protocol for sorting of the IgA positive and IgA negative bacteria using normal donor milk and stool. So it's a lovely overlap with the biobank work that we've got as well. So with that, I'm going to hand the podium over to Tina Chambers to fill you guys in a little bit on the clinical project work that we've been doing.
Thanks, Audrey. So most of you, I think, are familiar with this project. The focus of it is looking at antibiotic use in breastfeeding moms to determine the potential impact of commonly used antibiotic treatment during breastfeeding on infant outcomes. And the aims were to recruit 480 individuals who are currently breastfeeding and over a 3-year period of time, and to follow those individuals from baseline to determine those who started taking antibiotics in the ensuing period of time. These are all women who were recruited breastfeeding infants less than 4 months of age. And then once we identified an individual who was taking an antibiotic, to collect serial additional samples from those mothers and to select from the 480 at baseline infants that were matched on age and sex, and invite those matched individual moms also to give serial samples over that same term where they were not being treated with antibiotics. And then we had an additional sample for context of moms who were exclusively formula feeding. And so the aims were to look at the pharmacokinetics of commonly used antibiotics in breastfeeding, and then to look at the potential influence of antibiotic treatment while breastfeeding on milk composition, on human milk oligosaccharide expression in those milk samples, and then to look at microbiome and not only in the milk, but also in the mom and in the infant. And then finally to look at antibiotic treated compared to antibiotic not treated in terms of growth and development of the children. And so in terms of milestones, we're pretty much on target. We recruited 406 out of the 480 mothers that were the target in Southern California. And they've provided the baseline milk, skin, stool, and environmental samples. And we have reached our target in terms of the antibiotic exposed and matched antibiotic unexposed, who were identified as antibiotic treated or not, and they've consented and completed the serial collection of milk, skin, stool, and environmental samples over the course of treatment. We are following up, as stated, for the 1-year outcomes for those infants in both the exposed and unexposed groups. So far, for the 1-year follow-up, 58 out of that 80 have reached the 12 months of age or greater, at which point we are capturing medical records and abstracting growth parameters and indications of serious and non-serious infections. And then the mothers are completing age-appropriate ages and stages questionnaires to look at meeting developmental milestones in those groups. The group that we've had much more difficulty in recruiting is the formula-only moms. So we've been working through recruiting through general pediatrics using MyChart invitations, and have recruited, I believe we're up to about 8 now, but plan to recruit that group in the near future. We did have a supplement under the clinical project, and the purpose of this was to use bank samples in our human milk biorepository that already had been collected over the previous 10 years. So a little over 2,300 bank samples, milk samples from unique individuals in the milk biorepository were pulled, and those aliquots given to Dr. Tsunoda's lab for metabolomics. And I think she's going to talk a little bit about that. The sample analysis is complete in terms of doing the metabolomics, and then description of the outcomes in preparation. We did also propose and have access 300 bank samples from unique individuals who had exposure while breastfeeding to monoclonal antibody medications or SSRIs. And under that aim, Lars Bodie in our group is looking at HMO analysis with those 2 medication groups. The sample analysis has been completed, and the data analysis for the associations between HMOs and those specific medications is in progress with Dr. Bodie. And then we also propose to recruit 300 new participants through the Human Milk Biorepository, representing SSRI, monoclonal antibody exposed, and then a third group of kind of all comers. And with that, we proposed with those new participants to test our ability to be able to collect paired blood samples from the mother and the infant using the protocol that we use for self-collection in the home across the U.S. So we reviewed and tested several kits for this self-collection of blood from mother and infant, selected this TASO device, and then deployed that. So as part of this aim for those newly recruited 300 participants, we have completed that enrollment, and the mother-infant dyads in that group have provided not only the milk samples, but we've successfully been able to collect the blood spots stool, skin, and saliva samples from those moms and infants. And so we are kind of the data collection platform for our MPRINT CET. So we're really poised in this next several months to be able to do a lot of interesting analyses. So we have proposed also to develop a common analytical platform to look at a variety of different aspects of human milk. And we've met with Jeremiah Momper, who's part of our team as well, to start on that aim to develop the common platform. And he's beginning with samples with exposure to small molecule SSRIs to develop a broad spectrum panel that includes lipids, proteins, drug concentrations, and then he'll move on to other therapeutics after the SSRIs. And we expect results of that, at least initial foray, by summer. And then finally, under the supplement, we propose to partner formally as much as possible with other networks and particularly PTN. So we did arrange with PTN to assay 200 existing sertraline exposed milk samples from UCSD. The samples have been identified and have been sent to the vendor. These for the assays, these are to be compared to 50 existing Cuddle study sertraline samples. The planned FDA submission for the sertraline component of the Cuddle study, whenever that is, the idea is that could these 200 existing sertraline samples that were collected using a different protocol than Cuddle, not under clinic conditions, but under the mailed self-collection conditions, would they meet the criteria? And you heard Elizabeth Thompson talk about some of the approaches to looking at that. So we're waiting for those analyses to be completed. And then finally, working with our colleagues at Vanderbilt, we sent a subset of 50 existing sertraline exposed milk samples where Digna and her group sequenced maternal DNA from the milk samples. So we have the genome sequencing data that just was recently sent to us and are working with our computational biologist to analyze the data relative to sertraline dose and then levels in milk and how that might be influenced by maternal genotype. And I will pass this on. Actually, I'm going to say a word about the Milk Analytics Core before we go on to George. So that Lars Bodie, who couldn't be here today, leads the Milk Analytics Core under the UC San Diego Human Milk Institute that we talked a little bit about yesterday. The Analytics Core had all along had capability to do macronutrients and is doing this for our clinical project. They have added, as I mentioned yesterday, micronutrients to their array of analytics that they can perform. And this includes minerals and trace minerals, vitamins, both water and lipid soluble. And then they continue to do HMOs or oligosaccharide assays and bioactive proteins and peptides, including lactoferrin, osteopontin, adiponectin, calprotectin, and others. And they've added 2 new staff members to the Milk Analytics Core shown here. And they are adding abilities to be able to look at milk fat globules and proteases. We did have a symposium at our Human Milk Institute symposium that took place a couple of months ago. And at that, we did an MPRINT joint set of talks. And this was very well received. And we did want to announce that the Human Milk Institute will have its next annual symposium March 4th through 6th in 2025 in La Jolla. We hope that there will also be an MPRINT session at that symposium. And the format for this in the previous 2 years since HMI was established was all invited talks. The format for next year with additional time at the symposium is going to be an extra day. We'll also have submitted abstracts and poster sessions, so opportunities for team building, collaboration, and for trainees. All right, and I will pass this on to Dr. Liu.
Good morning. I'm going to give you an update on Project 2. My name is George Liu. I'm an infectious disease physician who does translational research. I'll be talking about the project. The title is The Impact of Ampicillin and Breast Milk Oligo on Infant Microbiome Immune Function. This is a talk talking on behalf of Victor Nize and our lab. The work is done primarily by two talented project scientists, Fatma Oskarian and Chiming Tsai, with the new addition of Emmy Suzuki, a GI fellow here. So what motivated our, which one was it? Green button. Sorry. This one. Green button. Okay, so what motivated the study was lots of antibiotics are given to neonates for preventing neonatal sepsis to the mom, to the infant, maybe. And when folks look at it, at the rate of potentially you have to give 200 doses, 1 to 200 doses to prevent one case of sepsis. Okay, so that's a lot of antibiotic. And folks have found more recently that there is a lot of potential long-term effect of antibiotic, and this is starting to be understood. So we wanted to understand this at a deeper level. So we are giving in this project antibiotic, ampicillin, to the mom, to the infant diet, and then trying to look at the microbiome and look at the metabolome, and then assessing the effect of this on the immune function in the infant, both innate and adaptive responses. So more specifically, what we have here are 3 ways of giving the antibiotic. We're giving it either to the mom before delivery. So this gets to the infant through placenta. We're giving it through lactation in the first couple of days of life to the mom, and this is transferred to the infant. We inject it directly into the infant. So then we assess what's happening. So from the last part cycle, one of the most salient finding we had was that the infant actually that received the antibiotic prepartum, postpartum through lactation, all gain weight. And this is significant if you look at it across the board. In every single case, there's a weight gain in association with the antibiotic, just a couple of doses. That was sufficient to do it. And this actually borne out to some pediatric study done in the past also that showed that courses antibiotic, even a course, 1 week course of amoxicillin can drive potential weight gain over time. So this is quite concerning about what happened. So we wanted to understand what is potential mechanism associated. This is weight gain at 21 days. Looking at, this is Shirley's data with Peter from looking at the metabolome bile acid. And of all the bile acid that was looked at, there's a lot of changes in the maternal system. But in the neonate, there's only a couple that were different. So if you look at it, one of them is an unknown bile acid. The other one is actually known, taurocolic acid. And there seemed to be an inverse relationship between weight gain and level of taurocolic acid. And looking at the literature, that's quite interesting. There's a PNAS paper that showed in there somewhere that they show that this correlation between increased weight gain and decreased level of taurocolic acid is also present in our study. And the way they found it was that overexpression of this bile sulfate hydrolase if they're overexpressed, this can lead to weight gain also. So we're trying to really point down to the mechanism right now as to how this goes about. This bile acid hydrolase is associated with lipid metabolism and weight gain directly. And this is quite interesting. In addition to that, as part of a project that we took on, we were looking at the effect of these antibiotics on innate function, how the neonate is able to clear GBS. And we actually found not a lot of difference. but on the adaptive branch, response to vaccine. We also noted quite an interesting find. And this is a pilot study where we gave the ampicillin through breast milk on day 1 and 2 postpartum. And then we vaccinated the pup in the first 7 days with hepatitis B vaccine or Prevnar, both of which are neonatal vaccine. And we looked at over time what's happened to their titer and plan to challenge them with strep pneumo. Here you see on the screen here that hepatitis B they didn't actually respond well in the first 2 weeks, so I can't say too much about that. But in the case of Prevnar, antibiotic administration actually block the response to the Prevnar at 2 weeks, although over time you see this recovering. What's interesting was in the recent literature in the last year or two or so, there's a paper published in Pediatrics that looked at 560 kids. Out of 556, 300 of them had on average about 5 courses of antibiotic. And when they looked at it, they found that those kids all have sub-levels of antibody made to the vaccine. And they followed over time, and they found that even by 9 to 12 months of age, several vaccines are at the level that's below what would be called protective level. So here it is. Antibiotic probably will have an effect on how well you fare with immunization. And we're also trying to pinpoint what this is happening. The bile acid itself is known to change some of these adaptive immune response through Th1, Th2, and 17 axis. And also it is known that the microbiome has such an effect also. What that mechanism is, it's unknown. It's not very clear right now, so we're hoping to be able to give you some answer next time. We're also trying to use multiple doses to see some of these effects and do the direct challenge, see the effect. Finally, I want to talk about a support pool project that we just started. The title is The Impact of Maternal Milk Cytokine on Infant Microbiome and Susceptibility to Enteral Colitis. This was a project that was inspired at the beginning of the Human Milk Institute at UCSD. And as you know, there's infant formula that's highly fortified in nutrient, but doesn't have a lot of immune component. And we know that human milk, when given to infants, they have a lot of benefit, include decreasing susceptibility to NEC, and so some report of BPDs and all that also improve with human milk. So what component of human milk is really affecting this and conferring some resistance? So to answer this type of question, we actually wanted to look at some of these immune component in milk. So what we have doing is a cross-fostering strategy where we take wild type pups, which is being fostered by the dams that are IL-6 knockout, IL-1 beta knockout, and some are pro-inflammatory, some are anti-inflammatory. And we want to then follow what's this effect on microbiome, metabolome, and also the susceptibility to enteral colitis, et cetera. Of course, the long-term goal is that can we not just fortify the milk with nutrient, but actually have an effect on immune component to make that a type of milk that has more benefit to the infant. So with that, I want to pass on to Shirley.
Good morning, everybody. I just wanted to summarize a few of the studies that we've been doing in our group in the MPRINT hub over the last year. So the first study was a study in combination with the Nizay and Liu labs that Sydney Thomas led. And this was, as George was explaining, where we gave ampicillin administration and looked at bile acid distribution as kind of a focus of our metabolomics analysis, and found some interesting bile acid changes in the moms. And as George mentioned, the the significant weight gain with the pups as well due to ampicillin. Next was the multi-omic analysis that I described in pretty good detail yesterday, and that was conducted by Sydney, Simone, and Simone. That project is getting ready for publication. Peter was a co-author on a project looking at bile acids in early life and its connection to islet cell autoimmunity and a potential connection of bile acids to diabetes progression. And so that paper is also in the process of getting published. And then 3 other studies I'd like to highlight that are in various forms of production. So Sydney had started this work, but Kine and Vincent, who are postdocs in our group, are completing this study. We had the good fortune of being a collaboration with a Gates Foundation funded project where these were mothers and infants that were recruited to this study in Bangladesh. And we have access to a bunch of samples from this cohort of dyads in Bangladesh. And we have currently been characterizing the bile acid profile of these of this cohort. We have an observational cohort of 55 mothers and infants that were just followed. And then another cohort of 205 dyads who received a probiotic of Lactobacillus plantarum. And we're currently analyzing that data to look at the impact of the probiotic on bile acids, as well as looking at the correlation between bile acids and adverse outcomes in the infants. And then we also have a continuing analysis of our murine microbiome study. And this is being done by Simone Zuffa and started by Sydney, where we're looking again at the metagenomics analysis of these fecal samples in our murine population that has been exposed to ampicillin. And then finally, we are looking at the initial analysis of the 2,500 breast milk samples that Tina had discussed, where we're looking at the metabolomics of these breast milk samples. And it's taken a little while because we're creating some interesting drug libraries to help us annotate the spectral, all the spectral peaks that we get to identify more of them and to be able to identify more drugs and drug metabolites. So Nina Zhao and with the help of Kine have been working on creating this drug library that I'm, very excited when this is going to be completed, because we're going to be able to see many more drugs and drug metabolites in any sample that we run through our untargeted metabolomics pipeline. And so I do have a little bit more information about that. Nina collated data from multiple sources to try to increase the number of spectra and increase our ability to annotate the number of drugs and drug metabolites in any of our samples. And so currently we're able to annotate over 5,000 drugs and drug metabolites with this drug library. And she's getting also this publication ready. And Kine did a lot of work to connect the metadata so that not only do we have information about the drugs and the drug metabolites, but we will also have information on the therapeutic class and the disease indication for these drugs and drug metabolites. And this is just a preliminary graph showing you all of the different compounds, mostly drugs, but also some food. We are also, I think, as I mentioned yesterday, we're working on a personal care product library as well, so that we'll be able to annotate a lot of the topical medications and products that people use when they're pregnant, for example, and does that get into the breast milk? So we will be working on that very extensively over the summer. And then I just want to mention the Pharmacometrics and Analytical Chemistry Core on behalf of Dr. Jeremiah Momper, who could not be here. Dr. Momper and Dr. Best run this core for our MPRINT group, and they basically have the ability to create targeted analyses for drugs. And, you know, Jeremiah has also expertise in cannabis. He runs a pretty large cannabis laboratory for the county of San Diego. And these are some current projects he's working on in collaboration with Tina with some of the breast milk samples. He's creating some assays specifically targeted for SSRIs. He also has been working on other opportunity and support pool projects, providing analytical expertise for these projects. And all of this work has resulted in some publications so far. So we did a collaborative publication back in 2023 with our entire group. And then Victor and Lars's group recently published about human milk oligosaccharides. And then finally, we have on bioRxiv our ampicillin project, and we have several more in various stages of publication. So with that, I'd like to thank you for your attention and for the MPRINT funding.
So we had a number of UCSD speakers. If anyone has questions and they want to go up and ask them before we take a break, you can catch us at the break. All right, if there's no questions, I think we're going to take a quick break. And Sarah back at 10:45, she says. All right.